My name is Paul Lincoln. I'm your host for Soil Matters, a podcast from the British Society of Soil Science, the professional body for soil scientists in the UK, and welcome to the fourth episode. Soil Matters connects scientific research with practical application. Today, we've left the studio, and I'm delighted to say that we are in Colt Park Meadows, a traditional hay meadow located in the Ingleborough National Nature Reserve in the Yorkshire Dales National Park. We're here to discuss the history and future of a scientific experiment, and to help us do so, we're meeting two of the people responsible for making this site a place of significance. Ben Sykes is the Executive Director of the Ecological Continuity Trust, and Richard Bardgett is Professor of Ecology at Lancaster University. The Ecological Continuity Trust is a corporate member of the British Society of Soil Science. Ben and Richard, can I offer you a warm welcome to Colt Park? Let's start with you, Ben. Tell us, why are we here in Colt Park?
Well, good morning, everyone. I should say that there can be few better places to spend a couple of hours on a summer's day than deep in the heart of the Yorkshire Dales in the famous Yorkshire Three Peaks country. But of course, that's not really why we're here. We're here because Colt Park Meadows is host to a long-term ecological field experiment, an important field experiment that's now into its fourth decade of operations, and an experiment, crucially, that features quite prominently soil science research. And that is why the gentleman on my right is here, and you'll be hearing from him very shortly on that.
And tell us a bit, what are we looking at? We're sitting here in a barn, but we've got this rather amazing view. Tell us a bit about what's on the horizon.
So we're in the barn at Natural England's headquarters on the Nature Reserve here, right in the heart of Three Peaks country, so just underneath Whernside, Ingleborough, Pennygent. And this is a classic limestone Yorkshire Dales scenery here. We're looking out across the Yorkshire Dales National Park to some of the foothills of the Three Peaks.
When you talk about a classic scenery, can you just spell out something? What should we be paying attention to?
So we're looking at scrub vegetation, we're looking at limestone escarpments, we're looking at streams in the valleys and rolling, undulating upland countryside.
And say a word about the walls that we can see.
Yes, that's one of the famous dry stone walls, and I think there must be thousands of miles of dry stone walls. Richard, you probably know better than I do, in the Yorkshire Dales National Park, and particularly round here. So very much a classic feature of the scenery, Paul, yeah.
Okay, now, before we start looking at the area in more detail, tell us a little bit about why the work of ECT is relevant to soil scientists.
ECT maintains a national register of over 80 long-term ecological field experiments of various types. And of those, 38 are currently active, including this one here at Colt Park, and undergoing treatments. But many of those experimental treatments, which are out here in the real world, are above ground. So they're applied to vegetation, they're applied to plots. And quite often the soil beneath those plots has lain undisturbed for literally decades. And some of the experiments have a strong soil science component, like Colt Park. Richard is both a card-carrying ecologist and a soil scientist, but many of them don't. And the charity that I direct, ECT, feels that this is very much an underused resource by the soil science community, and that these undisturbed soils, beneath a range of different treatments, including the do-nothing control treatment, is rich for exploitation for research purposes by soil scientists.
Okay, and tell us a bit about your particular job in the organisation.
So I'm the exec director of the Ecological Continuity Trust. I essentially run the charity with a board of 13 trustees and 4 other staff, most of which are part-time. So I manage board meetings, I manage exec committee meetings, I take care of a lot of the governance, but I also run our small grant scheme. I manage our own podcast programme. Most of our charitable objectives are based around science communication and the data arising from long-term ecological field experiments and trying to help that be translated to end users.
Why are long-term ecological experiments important?
Okay, a whole host of reasons, Paul, which I won't all list here, but I'll give you three key reasons I think why long-term experiments are important. Firstly, they are in a sense time machines. So when you have an experiment like Colt Park Meadows here that's been running for nearly 40 years, they look back through a valuable data set to see where we've come from. But the treatments often simulate land management interventions or climate scenarios of the future. So they also look forward to see where we're heading. So that's one of the first most important things about them. Secondly, they are long term. So ecological change can happen quite slowly. And some of the hardest questions in ecology are around what happens with long term change, how long does it take? What are the impacts of that for science and society? And so there are questions in ecology you can only answer by setting up a long-term experiment to tease out true cause and effect over decadal timescales.
What's your definition of a long-term experiment?
For the purpose of registration with ECT, a long-term experiment has to have been running for a minimum of 6 years. But on the register, the park grass experiment, for example, has been running for 175.
Oh, okay.
An experiment makes it onto ECT's register by running for a minimum of 6 years or having funding for 6 years into the future. Going back to the three reasons, Paul, the third reason, important reason why we need LTEs is that they can be repurposed. So if you have a long-term experiment like Colt Park that's been running, I think, 35 years precisely, you have a research platform that can be used to address new ecological drivers that were not conceived of when the experiment was first set up. So to give you an example, ash dieback, a 30-year agroforestry experiment like Loch Gall in Northern Ireland, we'd never heard of ash dieback, but there are ash trees in the agroforestry experimental plots at that site now being studied because of ash dieback. Microplastics has come onto the agenda. Acid rain came up in the 1980s, and those experiments that had been set up in the 50s and 60s then had a new research driver that came along. Having helped to preserve those experiments, those drivers could be examined. But if you'd lost the experiment after 10 years, you've lost the platform and you've lost the ability to research the new drivers that come along, those unexpected new drivers.
Great, thank you. Richard, let's turn to you. Tell us a bit about your job.
Well, I guess my official title is Professor of Ecology, based at Lancaster University, and I only recently moved back to Lancaster, which I might touch on later, in that prior to that I was at Manchester University for 13 years. And I guess as a professor, I have many different roles. I guess one of the things that we are challenged with, but also one of the great things about our job is that we do lots of different things. So we do research, we do teach, we do outreach work, and also in my own case, I do a lot of work related to advisory work for policy, for example.
And tell us about some of the key developments in your career in terms of soil science.
It's quite a difficult question, really, because I've had quite a lengthy career. I would sort of pull out a few. I think probably the most significant one for me was when I first started my PhD. I actually did a degree in soil science at Newcastle University. They're not very common, those kinds of degrees, but I did a degree in soil science. And I remember when I was finishing, the thing that really struck me was that we were taught lots about soil physics and chemistry, but what really interested me was the ecology of the soil. And I remember having a course at the time which was looking at the organisms that lived there, and that's what really fascinated me. And I remember in those days when you applied for a PhD, there was a poster put on the wall of the list of options that were available in different universities. And I saw one of them at Lancaster University, and it was on grazing effects on soil organisms in grasslands. And to me, it was perfect. I saw that and immediately decided I wanted to apply for that. I applied for it. Unfortunately, I took on the job. So that to me was a really key development in my career. I think the other one I would just mention is when I finished my PhD, I was, like a lot of people at that stage, very unsure about what I wanted to do. I thought I wanted to work in conservation, but I also wanted to do research. And I actually started working for the Nature Conservancy Council. So that's now Natural England, where we are based today. And at the time, I remember thinking, actually, what I really want to do is my own research. And again, I just saw a job advertised. In that case, it was at the Institute of Grassland and Environmental Research in Aberystwyth. I saw the job, I applied for it, and I got the job, and I became a soil ecologist. And to me, it was really the first opportunity for me to do my own independent research. So they're probably the two big developments, I'd say, in my career.
Great, thank you. Now, tell us about the history of this particular site, and also how long you and your colleagues have been working on experiments here.
Right. Well, again, it's a long time. The experiment was actually set up in 1989. And it was actually Roger Smith, who was at Newcastle University. He was actually one of my lecturers who set up the experiment at the time. And the primary focus then was about botanical diversity. It was all about how to restore botanical diversity within these kind of meadows, which previously had been managed with intensive agriculture. And then I came along in 1993. I kept in touch with Roger, who was one of my tutors, and he invited me along to come and look at the experiment. And I was interested in soils and the ecology of soils. And that's really when I started to get involved.
And also long-term ecological experiments. I mean, what's fascinating is that they clearly are very long-term, but why not just explain why it's so important to have this long-term perspective?
Ben touched on quite a few of these things before. I think the most important thing is that most ecological processes take time. If you have short experiments, 3 years, 4 years, you can miss the main changes that will occur in any particular system. And I think that that's particularly relevant here because we'll probably touch on this later when we go out in the field, but it actually took 14 years before we saw sizable effects of the management interventions on the botanical diversity in soils within this system. You know, if it would have stopped earlier, we would have missed the majority of those effects. And changes often happen rapidly. You know, there's a lull period as things are changing, then suddenly you get a transition. And I think one of the great things about this experiment is it's been running so long that we've been able to see those changes over time. So I think it allows that sort of long-term perspective. I'll just touch on a couple of other things. I think the other thing that's particularly valuable here and in other experiments is it's a demonstration. You know, like people want to see how, if you manage the land differently, what the outcome will be. And here we have, you know, a perfect demonstration of how long-term grassland management interventions can affect the botanical diversity, but also the soils. So it's a, you know, a real resource for demonstrating the benefits of different management options for.
How often would you visit the site?
For me, it varies from year to year. I mean, I've been working here since 1993. There will have been periods where I might have only come once a year or something like that. But generally, we've had active research programs on the way. So during the summer, at least, people, either myself or people who work for me, would have been here pretty much most weeks or in some cases for significant periods of time. So we've had a reasonably constant activity in terms of people working at the site.
Tell us a bit about how you've managed this particular site.
When it was first set up, it was actually Roger Smith, who I mentioned before, who managed the site initially. And it was also in strong association with what was then the Nature Conservancy Council and now Natural England. So it's actually Natural England who performed the majority of the management. But the way it's managed has changed quite considerably. I mean, Ben mentioned before about repurposing the experiment, and that's essentially what we've done, because initially it was all about botanical diversity. So the treatments were targeted at that. And the main things then were grazing, different types of grazing treatments. There was a seed addition treatment, there was a fertilizer treatment, and there was a farmyard manure treatment, and also cutting date treatment. So it's a very, very complex and large experiment. But over the years, as we've seen which interventions have the biggest impact on soils and vegetation, we've sort of honed the experiment in. So now we have sort of 4 key treatments. One is plus or minus fertilizer. One is plus or minus a seed mix for conservation purposes to increase diversity. We've got another which is targeted on particular plant species, in particular things like red clover, which we know has a big effect on the soils and the vegetation. And then we have farmyard manure as well. So we've sort of prioritized particular treatments over the years.
How often do you publish reports on this particular site?
We mainly publish research papers in journals. Because it's long-term work, we generally have experiments running, 3-4 years. So each of those might generate 1 or 2 publications every 2-3 years. So we've had a sort of constant stream of publications.
In response to your published papers, do you get people requesting that you vary the experiments or to carry out further experiments?
Not so much on the basis of the experiments. I think that the only time when that has really happened is when the experiment was first established. It was funded by DEFRA, and there was a close association with DEFRA and myself and Roger Smith in particular in terms of what the best treatments would be to actually address the questions that they were most interested in. So that time, the experiment was shaped quite significantly by policy. But I think since then, it's been really more about the funding that we've had and the kind of research questions that we've been addressing.
Because I suppose there is a certain, dare I say, luxury in having such a long-term experiment that you can review it, discuss it, review it again, publish.
Absolutely.
That you're part of presumably quite a wide scientific dialogue that's happening here.
Yeah, and I think that's one of the fantastic things here. I was just talking to Andrew, who's the warden here, and they'd just completed vegetation analysis of all the plots. And that was last done, I think, in 2019. But we've got vegetation data going right back to the start of the experiment. And similarly, soil carbon. We've been looking at soil carbon in these plots since about 1993. So again, we have long-term data that we can do. And just to give an example, we did just publish a paper recently. It was a postdoc who worked for me, Shangxi Liu, who was the lead author. And what we did then was draw on this vast amount of long-term data, which we pulled together to look at how all these different treatments have affected the overall functioning of these systems. And we couldn't have done that without that long-term perspective and data to allow us to collect and see how things have changed over time.
And let me ask you a question that you've touched on already, but how are you changing the ways in which you work as you engage with this particular site?
Probably in 2 ways. One, which is perhaps not such an obvious one, is technology. Right. So 40 years, things have changed. So when I first started, you could measure basic things about the biology of soil, for example, whereas now we have molecular techniques where we can look at the diversity, the functioning of those organisms in really complex ways. The other thing that's changed is that we've been using sensors, for example, to look at soil health. So we had a recent project where we were using different sensors in the plots to see whether we could sense changes in soil properties and relate that to the ground measures that we've been making about production and the fertility of the soil. So I think technology has changed and enabled us to address different questions in a more in-depth way. And I think the other thing that's changed is that as we've gone through the experiment, the kind of questions and environmental challenges have also changed. So it's gone from botanical diversity, then we shifted to carbon sequestration, and now we look at what we'd say is multifunctionality, which is a bit of a mouthful, but basically what it means is the capacity of soils or the system to deliver multiple services or benefits to people.
And what have been the most recent results of your work here?
Well, as I mentioned before, we recently published a paper which was based on data we collected over several years on a total of 26 different properties of soils and of vegetation. So we pulled all that data together and we did an analysis which basically showed that if you combine or increase the number of interventions, so in this case, if you have 4 different land use interventions combined, farmyard manure, seed mix, nitrogen fertilizer, and also legumes, you increase the functioning or the overall multifunctionality of the system. Whereas if you just use 1 intervention or even 2, you often get trade-offs. So, for example, if you add a small amount of fertilizer, you might get more yield, but you'll have fewer plant species and fewer pollinators. Whereas if you increase the number of interventions, you actually reduce those trade-offs and you increase the overall functioning of the system. And that includes things like carbon sequestration in the soil, nutrient retention in the soil, and the emission of greenhouse gases.
And let me ask you a question about climate emergency. In terms of this issue, which aspects of the research here do you think are most useful?
Yeah, well, climate-related topics became more prominent here in the sort of 2010 onwards, where we were funded by BBSLC at the time to look at how the different management interventions affect carbon sequestration and greenhouse gas emissions. And one of the things that we found was that benefits of biodiversity management seem to overflow in terms of benefits for the sequestration of carbon in soil. So in other words, the management interventions that were best for biodiversity, plant diversity, and also diversity of organisms in the soil were actually also the best for the sequestration of carbon and also minimizing greenhouse gases from the soil. And more recently, we've been looking at what I would call resilience, which is the capacity of soils and soil processes to resist and recover from drought or other climatic extremes. And again, we find these additional benefits of high biodiversity. The management interventions that have increased botanical or been beneficial for diversity seem to be the most resilient in terms of their ability to deal with climate extremes.
And let me ask you a completely unfair question. To what extent do you think the work here is having an impact on government policy?
Well, that is a difficult question. I mean, I think it's a lot like a lot of research. I think when it first started, it certainly informed recommendations in terms of how best to manage the land for botanical diversity. At that time, in the early 90s, there was a lot of emphasis on increasing botanical diversity in particular. And I think some of the results from here have been very helpful in terms of informing those decisions, particularly locally in this area where you get these herb-rich meadows. As it goes on, it's always really difficult to say this experiment here had this impact. I would say more that it's contributed among evidence from other systems as well to those overall policy decisions. I think one of the limitations with any experiment is that it's one field in one part of Yorkshire. How applicable is that? So I think it's like proof of concept almost, which adds to a weight of knowledge gained from a wide range of sites in terms of how to best manage the land for, say, ecosystem services.
I just spent the weekend in Bradford.
Yeah.
And the city centre has invested a huge amount of money and energy in creating new landscapes right in the heart of the city, replacing highway. And as an example of bringing nature into the city, I doubt if it can be bettered. And I just wonder again, to what extent work like this may have an impact on biodiversity within an urban setting.
I certainly think it can help and inform because, essentially what you get from this experiment and other experiments is recommendations on how to manipulate the plant community, for example, in a way that will deliver additional benefits. So I talked a lot about biodiversity, and actually we know it's not diversity per se, i.e. the number of species. What we do know is that certain plant species can have a disproportionate effect. So you can actually design potentially plant communities in a way based on their root systems, for example, which could either accelerate their restoration or bring additional benefits to the soil. So I think information from these kind of experiments can help fine-tune decisions about how best to restore systems in other environments as well.
And a word on the next 100 years for the site. What are your hopes for this particular piece of land?
Well, my biggest hope is that it continues. We might touch on that later about funding and that you need funding to keep these things going in the future. So I think in that sense, that's my hope is that we can continue to run the experiment because I think the benefits to biodiversity become stronger with time. And I think the benefits of these kind of experiments become stronger similarly with time. In terms of the science, there's two key areas we're focusing on. One is the actual diversity of organisms in the soil, in that most of what we've looked at, as I mentioned before, is more looking at groups of organisms and the functions they perform, whereas now we're using molecular tools to look at the actual diversity and complexity of those communities. and relating that to the diversity of organisms above ground. So we're now exploring more issues related to below ground diversity, which wasn't traditionally a main focus of this experiment. And I think the other one I'll just briefly mention is the one I touched on before about resilience. You might not think it looking out here, but droughts are even becoming more common in these parts of the world. And we're trying to understand, how can you best manage grassland systems in this case in a way which enables them to adapt to future increases in the intensity and frequency of these drought events. So that's quite a big focus of the work we're doing at the moment.
You've touched on funding. Are you optimistic about future funding for this site?
I am, yeah. I would say, I'm generally optimistic about most things, but I'd say that it's a challenge. Well, I think one of the biggest challenges you always face with long-term experiments is that people won't give you money to just continue to do the same thing. Yeah. And we've had to, over the years, come up with new proposals addressing new areas or new ideas to get the funding. Well, we've been successful in being able to do that, but it isn't without challenges. It relies on having the ideas, it relies on writing the grant proposals, and funding is also very difficult sometimes to achieve. I should say here that we've had tremendous support from Natural England, in that they maintain the actual running of the actual experimental treatments. And I think without that, and also the contributions from ECT, it would be a real struggle.
So actually, let's talk about some of those partnerships, because I think that's very interesting. Say a bit more about the partnership with Natural England in terms of who does what.
Well, Natural England, they own the land, and they always have owned the land. And we've always worked very closely with the wardens at the site, who've been incredibly supportive in terms of supporting the people who come here to do the experimental work. But I think the key thing that they've been able to help us with is access to the experiment, but also just maintaining the experimental plots, because they're here all the time. They help or take a key role in maintaining the actual experimental treatments. And without that help, that would be a major challenge for us, because we rely on generally short-term funding and bursts of funding. So we might have a period of a year in between grants where we simply don't have the money to be able to help support people to come to do the work. So they've been able to help us maintain the actual experiment.
And if I can turn to you, Ben, tell us a bit more about the relationship between yourselves and Natural England and this experiment.
The experiment is on ECT's national register. To come back to Richard's point about threats to experiments and funding, the two main reasons that ECT was established back in 2008 were because of the threats of closure that long-term experiments across the country were facing, either through lack of funding or particularly through lack of succession planning within academic institutions. And that's why continuity is ECT's middle name. One of our key raison d'etre, if you like, is that we are there to help smooth things over when funding dries up, or when a principal investigator retires or leaves or sadly passes away. We provide that bridge. And in that sense, we're unique. There's no other charity in the UK, or that we know of abroad, actually, that performs that role. What we can't do is provide the kind of BBSRC size grants that Richard was talking about. Those are the real bread and butter of researchers. So that's one of the research councils. Right. Yeah. So most experiments are kept going by DEFRA grants, Research Council grants, the sort of 3-year grants that a researcher uses to hire a postdoctoral researcher and some technical support and consumables. ECT's grant scheme is very small by comparison, but it's those small amounts of bridging funds that can help to keep an experiment like Colt Park going until new funding comes in or a new team comes in under a succession plan.
Obviously, we're going to have a look around the site in a minute, but in terms of your overall plans across the UK, what can we be looking forward to?
So at the moment, and for some time now, we've had experiments on our register across all 4 countries of the UK. And being on our register means that you have financial privilege to our small grant scheme. And there are many other gratis benefits in terms of profile raising and advocacy to government and policymakers around LTE's long-term experiments that ECT carries out. increasingly, we find we're adding experiments to the register each year at the landscape transformation scale. So, for example, Linbrianny in Wales, Glenfinglas in Scotland, the Wicken Fen vision grazing experiment in East Anglia. And the future drivers around landscape change seem to be indicating that more and more experiments are needed that look at the landscape scale. The plot scale remains very important for science. The landscape scale shows how things change at an ecosystem level in response to treatments and interventions, including the kind of things that are happening here on Colt Park, just at a larger scale. So I think that the future landscape scale transformation area is where ECT's efforts will be focused on in the near future. And we're, in partnership with the British Ecological Society, currently setting up a new resilient landscapes partnership with a number of government partners and a number of non-governmental organisations to help identify best practice, to signpost to science, and to create an information portal and build a community where interventions that work and interventions that don't work are shared and that we all learn together.
And are you optimistic about funding the work like that?
So a small charity like the ECT never seems to be more than 2 or 3 years from funding oblivion, and that's quite common for small charities, and certainly in the environmental sector. ECT only has 2 main sources of funding. One is private donations, and the other is grants that we can win from external charitable trusts and foundations and societies like, for example, the British Ecological Society.
And also, can I ask you, if you've got students who are interested in getting involved in your work, are there ways in which you can offer them placements?
Yes, we act as a facilitator in that respect. So we don't own the experiments and we don't own the land. But if a PhD researcher has a research idea, well, they can search our website for a suitable research site. Yeah. Alternatively, they can just pick up the phone to me, drop me an email and say, look, I have this research idea. Can you quickly identify a researcher like Richard and an experiment where this PhD research would work? And then we put them in touch with the researcher. We make sure that the research won't disrupt the integrity of the treatments in the experiment. And if it doesn't do that, there's often a route in. And that's an important role for us as a charity, because we want to see a wide number of academic researchers using experiments like Colt Park as research platforms to come and do soil science or to come and do above ground work that complements what's already going on there. And actually, when senior leadership teams in universities are having conversations about cuts, it can be very easy to close a long-term experiment that is just has one researcher on it. Whereas if you have several institutions collaborating, and there are more than one institution working at Colt Park here, it's much harder to justify closing down an experiment in a cuts conversation.
And in fact, what's really interesting is with such a long-term commitment, what you don't want is the kind of the short-term cycle of a 4 or 5 year parliament to have an adverse impact.
That's right. We've already touched on why long-term is so important and why if you stop something because you think nothing's happening, you may miss a crucial piece of science that may be critically important to an end user and landowner who wants to increase biodiversity on their land. And Richard mentioned that Colt Park took 14 years to show change. At the Buxton climate change experiment in Derbyshire, the climate change treatments that they were applying to the plots, I think, took 8 years to show any change. And Richard, please comment on this. What I think that shows is that the principle that biology doesn't waste energy. So plant communities sense their environment and they don't invest energy in evolution unless the signal they receive has some semblance of permanent change about it in the environment. And that's what climate change appears to be, sadly. And so that tipping point varies in different habitats and different climatic conditions. And only by running a long-term experiment for the sort of timescale like we have here at Colt Park, 35 years since 1989, can you identify when those things happen and then feed those, crucially, into models? In addition to the three important points I raised earlier, one of the things about long-term experiments is they feed into bigger environmental science models, data that comes from them, so that that data can be predictive.
I was going to raise another point, actually, which I think is not often appreciated, is how valuable these long-term experiments are for educational purposes as well. In that, just here, for example, we've had PhD students, master students, undergraduate project students from all over the world have been here and done their project work here. And I think increasingly, we need to make sure that to be an ecologist or an environmental scientist, you have to get out in the field and you have to observe patterns in nature and learn the species and learn about how systems change. And these kind of experiments provide a fantastic, natural laboratory to be able to do that.
Okay, thank you for that. Well, we've been sitting in this nice, warm, dry bar, and I think it's time for us to go for a walk. So let's have a look at the experiment.
We're just walking out into the meadow now. The actual experiment's just on the horizon over there, about 200 metres. You can see some fence posts. It doesn't look anything particularly spectacular at this time of year, but those fence posts that you can see in the distance, they're marking out the different blocks of the experiment. So within each block, we have all the different individual treatments repeated. So we have, in this case, 3 repeats of every single experimental treatment and all their different combinations. And I mean, in terms of the actual history of this site, before the experiment started, it was what we would call an agriculturally improved meadow, which are pretty prevalent in this particular area. It would be fertilised, raised, quite high intensities. It would receive manure additions quite regularly. So it would be relatively species poor. And at the time, there was a drive towards increasing the botanical diversity of these meadows to sort of restore them back to what would be considered a traditional herb-rich meadow. So we're walking up to one of the plots at the moment. I would say that this isn't necessarily the best time to see the treatments because the weather's been pretty grim recently. The plots have been hammered by rain, for example, and a lot of the flowering plants have shed their flowers as a result. But essentially,
each plot is how large?
So we walk up now. You can see each plot is basically marked out by these canes. So it's about 2 meters by 3 meters each plot, and then we have 3 of those plots replicated in different blocks. And within each of these areas, we have quite a large number of plots for each of the individual treatments. So as I mentioned before, that's with or without fertiliser. It's not a lot of fertiliser, actually. It's a very, very small application of fertiliser. Farmyard manure would be another treatment. We have a seed mix that's added, which is a mixture of seeds which are representative of the herb-rich meadows that you'd find in this area. And then we also have a treatment which is red clover. I'm just sort of looking for some red clover at the moment. You can just see some here. So this is Trifolium pratense. And one of the reasons why we have that treatment, we added it some years ago now, was because we found that that particular species seemed to have a disproportionate effect on the soils, including their capacity to store carbon. It actually increased the amount of carbon contained in the soil. So here we have each of those individual treatments plus every single combination that's possible of those treatments.
And how do you make sure that you don't contaminate adjacent plots?
Well, in terms of the actual treatments like fertiliser, farmyard manure, that's relatively straightforward. I think that the thing that's happened over the years is that the plant communities have changed, largely because of dispersal between the individual plots. So as some plots have been particularly successful, the plant species within those have moved to the different plots, and in some cases they've been able to colonise. So the vegetation has changed as a result, but you can still see quite dramatic differences. If you look at these plots here, they're just green. There's a lot of docks in there, for example, and they're very typical of the kind of plots which would be representative of more intensive management. So fertiliser addition, farmyard manure addition, for example. So they're very productive for yield, but there you can see there are very low botanical diversity. One thing I also didn't mention before was that the initial goal of this experiment was to recreate what would be a typical herb-rich meadow from this area. In scientific terms, that's called an MG3, which is an Anthoxantha moderatum meadow, which are quite prevalent in this area. And prior to that, when it started, it would be more like that one there, which is just a productive, relatively productive, species-poor meadow.
Okay, shall we walk
on
the edge a bit?
I mean, one of the challenges that we always have in these kind of experiments as well is infrastructure, in that we use a lot of equipment to measure things like greenhouse gas fluxes from the soil. We also sample the soils regularly, so one of the things we have to be really careful about is that we don't want to damage the plots too much, so we have to be very careful about making sure the samples that we do take we really do need, and we take into account previous sampling that we've done at the sites.
And do you want to say a word about the setting, because there's actually a rather dramatic
view that
we've got here.
I mean, Ben touched on this before. So we're in the meadows. I mean, you can't see much afar, but as was mentioned before, you're right in between what would be the three peaks. So Ingleborough is to the west, up to the north is Whernside. You can just about see the top, but it's got a cap on
in the clouds.
And then Penagent's over in the to the east there. So you're right in the heart of the three peaks, and this particular meadow is relatively uniform. So that's one of the reasons why it would be a meadow, because it's some of the more fertile land that historically could be managed to produce hay effectively, and also for grazing during the autumn and spring, whereas surrounded by us is more steeper ground, hill ground, lower productivity ground, which is of less value, I guess, in terms of producing hay and fodder. So this land around us will be used more for sheep grazing, cattle grazing.
And do you know how this particular site was chosen in the very first place?
I think probably Roger Smith, who set the experiment up, worked very closely with the Nature Conservancy Council, and I suspect he would have scouted plots and sites around here, and then this one came up as probably the best one.
Brilliant, thank you.
I think, yes, it might just be worth saying something about the different habitats that long-term experiments across the UK are cited on. Of those 38 currently active experiments on ECT's National Register that I mentioned earlier, quite a lot are grassland, and this is an example of limestone grassland here in the Yorkshire Dales. But there are also experiments on the register on peatlands, on woodlands of various types, on coastal sand dunes, and understanding the science of, for example, biodiversity in different habitats in different locations around the UK all feeds into the bigger science picture that Richard was talking about when you asked him about the impact specifically of Colt Park in policymaking. Collectively, these LTEs feed into a very rich body of science that tells us a lot about how we might manage different habitats in different locations in the UK in the future.
So we're just walking across the site, and I think a really interesting question is to ask about the way in which the different treatments have worked over the past few years.
So we're just coming up to one of the other plots here, and if you look across them, the big difference that you will see visually is height of the vegetation and also the diversity of the plants. So you can see directly in front of us of these plots here, which are very productive, but they're relatively species poor. So this would be a plot that's receiving fertiliser and farmyard manure. So it would be the historic treatment that we were trying to change when we first set up the experiment. And then next to it there's one which is more botanically rich. You can see all the flowering plants within it, the daisies, the ranunculus, and also there's the red clover. And I guess one of the things I mentioned previously that we've found in this experiment is that those treatment combinations that are best for botanical diversity, or the diversity of pollinators, for example, seem to be also the best in terms of processes that operate in the soil. And one of the things we've done recently is look at a broad range of functions that the soil provides, the kind of functions that would underpin, say, soil health from a biological, physical, and also a chemical perspective. So we've measured a wide range of properties. And what we find, for example, is the treatments that have seed mixtures added, farmyard manure added, and red clover, and also some farmyard manure, they are the most effective in terms of soil health generally. So they have improvements in soil physical properties, the structure of the soil is better, there's improvements in terms of their capacity to cycle nutrients and retain nutrients, but also, as I mentioned before, they also see benefits in terms of carbon sequestration and the build-up and accumulation of carbon in the soil. So I don't really like saying win-win strategies because there's always a trade-off somewhere down the line. But essentially, it seems by combining different treatments together, which bring benefits for biodiversity, they also seem to lead to additional benefits for the health of the soil.
That is fascinating. Now, if some of our listeners want to follow up some of the work that you've been doing, what's the best place for them to?
Well, people can contact me directly if they're interested. On the website, the ECT website, all the publications are listed, or most of the publications will be there listed related to it. And the paper I just mentioned was only recently published earlier this year in a journal called Nature Communications by an author called Shangxi Liu. And that included a wide range of different people who've been involved in the experiment over the years.
Now, the mists are swirling, so I think we're going to walk back to the barn quite soon. But before we do so, I'm really interested to know a little bit about the history of your organisation, and Ben, and tell us just about what led to its creation.
Yes, so the ECT was founded in 2008, so we've only been going 17 years. But it came into being through conversations that started at the turn of the millennium within a group of applied ecologists who included people like Jerry Tallowin, Jonathan Silvertown, Carly Stevens, Mike Moorcroft, for example. And those ecologists were lamenting the apparent loss of long-term experiments around the UK, hand over fist, due largely to two threats that I alluded to earlier. One is lack of funding, and two is lack of succession planning when researchers move on. And so they were looking for an organisational model to champion, support and maintain long-term experiments and preserve them whenever they faced threats. And so they set up the ECT in 2008, originally with an endowment model in mind, but that didn't come to pass largely because of the financial crash. But the charity got started with a small grant from the British Ecological Society and has grown from there.
And if people are interested in looking at new sites and doing experiments, should they contact you in order to discuss the possibility of you supporting them?
Yes, so not only do we continue to add experiments to the register, and you can see pages for all those experiments, as Richard said, on our website, but we do from time to time get involved in conversations with the researchers who are looking to set up new experiments. And if those experiments have more than 6 years funding into the future, they can become part of ECT's national network. And we have ecologists on our board of trustees who can help provide advice on design and setup and how to make data available to the wider community.
Great. Richard, Ben, thank you very much for showing me around today. If you'd like to find out more about this work, have a look at the podcast page on the BSSS website, and I look forward to being with you again for the next episode. Thank you.
Hi, my name is Professor Jack Hannam, and I'm a soil scientist, and I'm past president of the British Society of Soil Science. Did you know the health of our soil affects everything? Our food, our climate, our health, and our future. Formed in 1947, the British Society of Soil Science is a professional membership body dedicated to protecting, improving, and discovering new knowledge about the foundation of our planet, soil. From developing cutting-edge research, engaging with international and domestic governments through science-led policy and advocacy, to publishing in leading sector journals, and accessing extensive educational resources, our members are given the tools to be at the forefront of shaping a sustainable future. Our members are central to making a real impact and facilitating lasting change. Through access to our extensive networks, they could connect with experts and drive the conversation on soil health worldwide. Join a passionate community committed to tackling the future generational climate, biodiversity and food security challenges. Don't wait, get involved today, because soil is where it all begins, and soil matters now more than ever. Find out more at soils.org.uk.